Okay, I think we should start. Welcome everyone to WACBE webinars, our distinguished biomedical engineering webinar series hosted by the World Association for Chinese Biomedical Engineers. So my name is Siti Lim and I'm a NUSS professor from the National University of Singapore, and it's my honor to chair this webinar. So in terms of the webinar format today, our speaker will present for approximately 40 minutes, and we will leave about 15 to 20 minutes at the end for Q&A. So throughout the presentation, please use the Q&A feature at the bottom of your screen to submit your questions. We will try to answer as many questions as possible during the Q&A session. So today we are very excited to welcome Professor Leong. So Professor Leong is the Samuel Y. Shen Professor of Biomedical Engineering at Columbia University. He received his Ph.D. in Chemical Engineering from the University of Pennsylvania. So after serving as faculty in the Department of Biomedical Engineering at the Johns Hopkins School of Medicine for almost 20 years, he moved to Duke University in 2006 to focus on the design of nanostructure for therapeutic applications. After moving to Columbia University in September 2014, he continues to work on nanoparticle-mediated non-viral gene delivery and gene editing. And he has published more than 400 peer-reviewed articles and also hold more than 60 issued patents. He has won many awards, and I'll just mention a couple. One is the Distinguished Scientist Award of the International Journal of Nanomedicine, and the Clemson Award for Applied Research of the Society for Biomaterials, as well as the Lifetime Achievement Award of the CASNN. He is currently the Editor-in-Chief of Biomaterials, a member of Academia Sinica, as well as elected fellow of the US National Academy of Inventors, National Academy of Engineering, and also the National Academy of Medicine. So he's a very distinguished researcher. Actually, I know Kam for almost 20 years. When I first started as a young assistant professor in Singapore, he was our distinguished visiting professor, as well as he came quite often because of the Singapore Johns Hopkins program, and also thereafter the NUS. And he's also someone I always look up to, of what it means to be an excellent biomedical engineer. So I now pass this time to Kam.
Thank you, Siti. Very kind and nice. It's really a pleasure to be sharing the work with you guys. It's a little bit difficult for me to present over Zoom, so I made a recording so that I thought it would go a little bit smoother, and also the timing would be better. But I will be here and I will be handling all the questions and so on. So it's semi-recorded and semi-live. So again, thank you. And because of time, I will just get started. I hope there won't be any echo, and I hope this will work. Again, all my colleagues, all my friends, thank you for coming, particularly for my good friends from the West Coast. Sorry to make you wake up so early, and also in Asia, you are late. So I hope this will be worth your while. Thank you again. Hello, thank you for coming to the seminar. It is a pleasure and honor to share with you our work on designing biomaterials to modulate inflammation. Inflammation is ubiquitous in our body. It plays an important role in responding to danger signals arising from damage to our body and in restoring homeostasis. Controlling the inflammatory response is a major strategy in managing diseases such as cancer, autoimmunity, and wound healing. Now, conventional drug therapies using low molecular weight drugs are the norm in tackling inflammation, but biomaterials can play an important role. Through drug delivery, biomaterials can enhance the efficacy of anti-inflammatory drugs. Biomaterials can also be applied in an inhibitory mode, for example, using sulfate polymers to block cell adhesion molecules to prevent the adhesive interactions of leukocytes and endothelial cells in initiating inflammatory responses. Biomaterials can also be applied in a scavenging mode to remove the pro-inflammatory factors such as reactive oxygen and nitrogen species and cell-free DNA that act as damage-associated molecular patterns, the DAMs. In this talk, I will focus on this particular aspect, on the use of biomaterials to scavenge the DAM molecules. Dead and apoptotic cells release their intracellular content into the intracellular space in the form of nucleic acids. These nucleic acids can be associated with lipids, proteins, and vesicles, and so on. These molecular patterns can be sensed by immune cells such as macrophages by the toll-like receptors, which can then activate the inflammatory network, leading to secretion of cytokines. That is a good thing, part of the defense mechanism of our body. However, when the inflammatory network is inappropriate or overactivated, it could lead to many inflammatory diseases, such as the one shown in here. So pharmaceutical companies have been developing drugs to specifically block this activation. However, there are two drawbacks. There are many TLRs, and they each recognize a particular molecular pattern. First, one may not always know the exact pathogenic molecular pattern, and there may be multiple molecular patterns that are causing the inflammation. Second, these pathways are redundant. Blocking one particular pathway may not be efficient in inhibiting the inflammatory response. When I was at Duke, Jay Woolley and Bruce Salinger proposed the idea of binding the DAM molecules as a way to inhibit the inflammation activation upstream. We have tested this scavenging concept in a number of different models and showed therapeutic effect. After I moved to Columbia, I have continued to work on this approach, and today I will describe the most recent work on sepsis. But let me show you quickly what has been done so far. Because most of these DAM molecules are nucleic acid in nature, highly negatively charged, we tested a panel of polycalines that we have been using for non-viral gene delivery. The one at the bottom here is developed by Mark Davis at Caltech. The one with the highest DNA binding affinity is this cationic dendrimer, the polyamidoamine generation 3. So this is kind of our standard, and we will be coming back to it for the rest of the talk. This is the very first study done in an acute liver injury model induced by pathogenic DNA or RNA. Without treatment, all the animals die in one day. With the administration of the polycalines, some of the animals could survive. This is a somewhat artificial model, but it is a proof of concept that this scavenging concept in blocking inflammation could work. There are inflammatory diseases that are localized, so we fabricated cationic microparticles that are 5 microns or bigger in size, so they won't be easily internalized by cells, and apply them in a local manner. In the TMJ inflammation model, we show that these cationic microparticles could reduce the damage in the cartilage and bone of the TMJ. In another configuration, we tested the scavenging concept in a trauma situation, which can often lead to uncontrolled immunothrombosis and death. This time, the scavenging is in the configuration of a membrane, and apply in an ex vivo manner. We show that the serum of trauma patients will activate the TLR pathways. In a transplant situation, there can be a high level of DAM molecules. In this heart transplant model, treatment of the donor heart with the cationic membrane would prevent occlusive thrombosis and death. So far, I have shown you the three configurations of the scavengers: soluble polycation, cationic microparticle, and cationic membrane. Now I would like to show you next the nanoparticulate scavenger. In this case, we test the concept in a rheumatoid arthritis model. RA has both genetic and environmental factors, and both are related to TLR activation. For example, microorganism-associated molecular peptides, the MEM, can activate the TLR producing pro-inflammatory cytokines and chemokines and MMPs, causing tissue damage. Cell death producing more DAMs, kind of a feed-forward cycle. We ask if scavenging of the pro-inflammatory factors can alleviate the disease progression. We constructed the cationic nanoparticle from this diblock copolymer with the PLGA as the core and this cationic PDMA as the shell. We compared the cationic nanoparticle with the soluble PMDA, PDMA in the study. In one of the most widely used RA model, induced by collagen II, symptoms would first appear around 2 weeks. That's when we began treatment. But we also included a group that began treatment 1 week later to mimic the clinical situation better. We did a thorough characterization of the disease parameters. MRI showed there was less tissue swelling in the ankle and the foot. Clinical scoring also showed less joint swelling. Micro-CT also showed less bone damage. Let us focus on these 6 groups: normal, model, soluble polymer, cationic nanoparticles with roughly half the amount of the soluble polycation in early treatment, double the dose in early treatment, and high dose cationic nanoparticles in late treatment. In all cases, there was a reduction in bone damage in the experimental groups, which corroborated with the bone density measurement. We did many mechanistic studies to understand how this scavenger works and why the nanoparticles work better than the soluble polycations. I will save those explanations in the sepsis model. In this experiment, we look at the cell-free DNA level in circulation. In the control group, the cell-free DNA level was about 40 microgram per ml. In the RA model, the level was significantly higher. The soluble polycations could not lower the level, but the cationic nanoparticles could efficiently. The cationic nanoparticle was not as efficient in the knee joint, but it still lowered the cell-free DNA level. And this was corroborated with the lower levels of pro-inflammatory cytokines, chemokines, and enzymes in the knee joint. In the rotating cage evaluation, normal rats could handle up to a speed of 8 RPM. Model rats could hardly do it. probably because of the damage in the joint and the pain. These treated rats could do quite well. This is the best guy in the group. Not all rats were this good. However, on average, there was a partial functional recovery in movement of the treated animals. Let me move to the last animal model. Sepsis is a significant healthcare problem, the third leading cause of death in the US. It evolves from a local infection to systemic inflammation to multiple organ damage and in some cases to death. We first made sure the scavenging approach would make sense by measuring the cell-free DNA level of septic patients. Indeed, the level is significantly higher. The serum of septic patients would activate TLR9. Treatment of the patient's serum by PAMM, the first soluble polycation I showed you previously, would inhibit the TLR activation, which is reflected downstream in a lower secretion of the pro-inflammatory cytokine TNF-alpha. Then we set up the sepsis model, the most widely used preclinical model, the cecal ligation and puncture model, which is the perforation of the cecum, allowing the release of fecal material into the peritoneal cavity to generate an immune response induced by polymicrobial infection. We vary the distance of ligation to create different degrees of sepsis, and we focus the studies on the severe case. Without treatment, all the animals would die within 3 days. IP injection of the soluble polycation would improve the survival to 40%, and this was as good as the drug used in the clinic in China. And this was corroborated by the lowering of TNF-alpha, IL-6, and MCP1 in circulation. Mechanistic studies show that the scavenging inhibited the inflammation through the TLR9, MyD88, and a NF-κB signaling pathway, which led to the reversal of M1 polarization of the peritoneal macrophages. Like in the rheumatoid arthritis studies, we then designed a nanoparticulate scavenger to see if it would have a better therapeutic effect than the soluble scavenger as we have observed. This time, we picked the mesoporous silicate nanoparticle. Like in the rheumatoid arthritis study, we then designed a nanoparticulate scavenger to see if it would have a better therapeutic effect than the soluble scavenger as we have observed previously. This time we picked the mesoporous silicate nanoparticle as the core because the porous structure of MSN provides a high surface area for functionalization. And we could incorporate a disulfide bond into the MSN network, making it biodegradable as shown in here. We grafted PEI onto the MSN, 2 molecular weight. The company used 25 kilodalton branched PEI, which is quite toxic, and a linear 800 molecular weight that is much less toxic. Both nanoparticles are less toxic than the soluble PEI 25K and still could bind DNA reasonably well. Applied to the severe sepsis model, we could see a slight improvement over the soluble PAMM from 40% to 50%. The survival performance correlated with the cellular studies and the cytokine levels, which in turn correlated with the DNA binding efficiency of these scavengers. It also matched the reduction in damage to the lung, liver, kidney, spleen, and heart. The clinical chemistry was also better with the nanoparticulate scavenger than the soluble polycalines. The reason that the nanoparticulate scavenger performed than the soluble counterpart is twofold. One, TLR9 is located in the endosomal compartment. Nanoparticles naturally traffic to the endosomal compartment. We can quantitatively show that the amount and duration of the cationic nanoparticle in the endosomal compartment is higher than that of the soluble polycation. At the tissue level, the nanoparticle also show a higher accumulation in the inflamed tissue, particularly the colon. This is so because nanomaterials can target an inflammatory microenvironment through the enhanced permeation retention effect, or transcytosis, or a phenomenon called nanomaterials-induced endothelial leakage. Nanoparticulate scavenger has also an unique advantage because it can also be used as a drug carrier. Not only can it remove the negative factors, but it can also deliver a therapeutic agent at the same time, like a therapeutic carrier. We are applying this concept to cancer nanomedicine right now. So, in summary, the scavengers can be applied in different configurations, nano, micro, macro, to address different therapeutic needs. There are many questions we have not answered yet. What exactly is being scavenged? What other structures can serve as scavengers? What happened to the scavenged stem molecules? And what is the optimal modulation of information in terms of magnitude and also in terms of timing, which is very important. Early information may be good for sepsis or cancer therapy, but chronic inflammation would not be good. So what is the optimal? So there is a lot of science that can be done and should be done. With that, thank you. I would also like to thank my students, fellows, and collaborators, and I will be happy to answer any questions. So, thank you. Thank you for the time. And
Yeah.
concise, I would be happy to answer questions if there's opportunity.
Okay, thank you so much, Prof. Leong. Can you unshare your screen?
Yes.
First? Okay. Okay, so thank you very much, Prof. Leong. We will, before we begin the Q&A, I would like to call your attention to our next WACBE webinar on Saturday, February the 27th, where we will welcome Professor Yu Po Fang from Beihang University, where he will be presenting on biomimic design of biodegradable implants. As you can see on this slide, we have an exciting speaker lineup, so we hope that you can continue to join us for this webinars. I would also like to announce the 10th WACBE World Congress on Bioengineering, which will take place in 2022 at the SASTEC Conference Center in Shenzhen, Guangdong, China. Details for that event will be announced soon. So please follow Columbia BME on social media to receive the latest update on our webinar, our WACBE webinars. You can see their social media information on this slide. And you can also learn more about WACBE by visiting wacbe.net. Okay, so I think we have several questions to get through, and so we'll try to answer them.
I could also see it, the first one.
Okay,
so
okay.
Answer that one.
Yeah.
The question relates to the physical properties of the nanoparticles about size. This is for Tom. So the one that we use in the rheumatoid arthritis study was around 50 nanometer. Of course, there would be aggregation in vivo. However, that's the information we do not have. In vitro, we study the size only in buffer. We don't do it in plasma. So that may not mimic the situation well in vivo. The size distribution of that PDMA, PLGA, diblock copolymer, those nanoparticles, the size distribution, I do not have the figure off the top of my head, but it's quite good. The MSN was around the same size because we do want to make them small. They would be under 100 nanometer. We would like them to be small so that they could accumulate in the inflamed tissue better. But that's a good question. I really would not know in vivo what kind of aggregation we would be seeing. I could only say that there is definitely preferential accumulation of these nanoparticles in the inflamed tissues in 3 different models we have studied now.
Okay, I think there's also a second question from Zhang Yiming. So if the goal is to take out inflammation-inducing molecules by scavenging, I imagine you will inject into the bloodstream. I think that's what it means. But how do you take them out? Or what's after taking up these molecules? Do we assume they will just degrade and cause no harm?
Yes, that is a very good question. So is in the form of nanoparticle, it was administered either IV or IP intraperitoneally. And that was one of the major questions I mentioned. I do not have an answer yet, and that is a key question. What happened to the scavenged stem molecules? Now, the Sepsis study, that's why I moved to a biodegradable nanoparticle, so that they could be clear from the body, so it would be more biocompatible in the long run. What happened to the dam, the scavenged dam? We do not have an answer. The wishful thinking is, once they are bound, absorbed on the nanoparticle, the enzymes in the body will continue to break down those dam molecules so that they will no longer be recognizable by the immune cells, by the toll-like receptors. But we do not have evidence of that. We do have evidence that these scavenged nanoparticles, they do not re-enter the immune cells. That seems to be the case. Not very rigorous yet. I cannot rule out all the possibility, but that is one major question that we need to answer right now.
Okay,
so thank you.
Yeah, there's a question from Barbara Chan. So she's asking, in slide 24, you show the cationic biodegradable MSN, which degraded in 3 days. Is this in vitro degradation in buffer, or was degradation in vivo even faster?
Thank you. That image was, of course, only done in vitro only, and also in a bioreductive environment in the presence of glutathione. Now, in vivo, it could be slower or faster, depending on how much enzyme, pH, intracellularly would also depend on the glutathione level. So I would expect in an inflammatory microenvironment, these nanoparticles could break down quite quickly. Days.
Okay.
Yeah.
Right, thank you. So there's a question by Wang Yaodong. Will the amount of cfDNA scavenged if affect the clearance of the nanoparticles? How does the nanoparticle alter the toxicity of PEI?
Okay. Yadong, is this the guy from Cornell? Yes. Okay. Now, again, the core of the nanoparticle is biodegradable. So with time, it could be cleared either through the renal system or through a cellular mechanism. And the DAM molecules would be broken down by the enzymes further. That will happen. Now, how does the nanoparticulate form alter the toxicity of PEI? That's also a very good question, but definitely in all the studies we have done, the toxicity is lower. It could mean the cellular uptake mechanism is different, so that it is less toxic. But you probably know in the non-viral gene delivery field, the molecular mechanism of the toxicity of these hundreds of polycalines have not been studied systematically. We still do not have a clear picture. Which aspects of the polycalines are causing the toxicity? So I do not have a good handle on that. Other than the fact that this cationic moiety is interacting with the cell or with the plasma membrane in a different manner, making them less toxic.
Okay, thanks. Okay, so there's a question from Li Shi. Li Shi is from the Chinese University of Hong Kong.
Okay.
Okay, he said, I really learned a lot and have a question. Since most materials or their degradation products may also lead to extra inflammation, how to choose the suitable component and the dose for in vivo study?
Yes, that is a very, very good question. Now, I've been trying for years, many, many years, talk to the Chinese colleagues that I know. Show me the Chinese medicines that have a lot of primary amino groups. I would like to formulate them into a cationic nanostructures that they can be broken down into a therapeutic moiety. But I've actually asked also a PhD student to do a study on that, but we do not have that yet. Is a compromise that is difficult to bridge. That is, we need this cationic moiety to bind the highly negative charge nucleic acid. But polycalines by nature, probably because of the way they interact with the plasma membrane of cells and so on, they are toxic. So we just have to find something that has a reasonable toxicity value and still can be therapeutic.
Okay, there's one question for Yoka Chokalova. So the question is, do you anticipate they could be used for skin or mucosa wound healing?
Yes, any inflammatory situation, I think we would have a chance to exert a positive effect. We published a study on that that was not as thorough as we would like, but that was published in Macromolecules, and I could send you the reference if you send me a message.
Mm, yeah, I actually have a follow-up question on this because I think you mentioned just now about electrospun cationic membrane. Would
Yes,
that be one of the better, uh, or be used for skin or for wound healing, being more
yes,
a membrane
I,
structure?
that is being done right now. Yes.
Okay. And
You,
I think I forgot what the material is. There's a material that you use. I mean, your slide, I think there's one slide you mentioned about this membrane, this
right.
electrospun membrane.
So in that study, because it's used ex vivo, it doesn't go inside to the body. So
Right. Yeah.
biostable is even better. So we have tried many different polymers. So for example, even polystyrene would work. That is, I made the nanofibers scaffold with polystyrene, and then I do chemistry to graft a polycation onto the surface. That actually would work well.
I see. Yeah. So, okay. So there's a question about Kainan An. So it say,
And,
Cam,
and
outstanding
right.
lecture. Thanks. General question. Please explain again the mechanism that nanoparticulate is better than conventional soluble ones. This might be advantage. Any disadvantage of such approach?
Okay. Thank you, Andy. That's a very good question, and it's a crucial question. So let me answer the disadvantage first. In terms of penetration in certain tissue, soluble polycation would have an advantage over nanoparticles. There was a good question earlier, the first one asked, would these nanoparticles aggregate in vivo? That's likely to happen. If they aggregate a lot, then they may not be able to reach the inflamed tissue like we would hope to be. But in most of the study, as I said, we did see a more preferential accumulation of the nanoparticles. So, but that is one potential disadvantage of the nanoparticle compared to the soluble counterpart. Now, having said that, then the nanoparticle has one major advantage in that it will go into the endosomal compartment efficiently. In gene and drug delivery application, we don't want that. But in this scavenging application, that's good because the TLR9 receptor is primarily located in the endosomal membrane. And TLR9 is one of the most prominent receptor sensor that is in this inflammatory network activation. So nanoparticle would be able to target the TLR9 better. The other major advantage we are spending a lot of effort on is that my nanoparticulate scavenger can also serve as a drug carrier. I can also encapsulate drugs in it. So now what we are doing is to try to advocate this push-pull effect that I remove the negative factors, but at the same time, I deliver a therapeutic agent at the same time.
Yeah. Okay. So there's a follow-up question from Yiming Zhang. I think it's a general question. So it's asking, given the data you have shown here, what do you envision the next steps to be in your lab?
So Ed Guo is going to answer this question live, right, Ed? Okay. So, as I said, in the very last slide, we really like to gain a handle on the mechanistic understanding. The four major questions I ask: what is being scavenged? What other structures can be used? What happened to scavenge them? The other major question, which is difficult to answer, what is the optimal scavenging? That is, the information is not all bad. It is good. Just too high is not good, and then the timing is also crucial. So would like to get a better handle on that. The other major direction, as I just said, that we will be going next is to also deliver drugs at the same time. So using this as a therapeutic carrier. That is, this nanoparticle alone is already therapeutic. Now I can add a synergistic effect by encapsulating drugs in it.
Can I? Hi, Cam. That's very good point, because we just had a model out for how the cationides nanoparticle ranging from 20 to 100 nanometer has a very much like efficient for drug delivery across endothelium, like from exocytosis, at least 100 times. Very good point. Yeah, I really like it. Before we are worried about if cationides particle is toxicity. Like you said, you can neutralize this toxic effect. So that's very nice. Yeah, great. Right, we can work on that. Great.
Sure, Thank you.
Okay, so we have a question from Li Jin, one of my colleagues from NUS here in Singapore. So he said, thanks for the great talk. This is Li Jin from NUS. Could you share something on the nanoparticle design rationales? What characteristics like size, stability, circulation lifetime, degradation, etc. are most critical for their functionalities?
I think a little bit smaller would be good. So under 100 nanometer, I would be happy. Then you will have a good chance of going into the inflamed tissue. In terms of biodegradability, that would vary with therapeutic applications. Some, I could imagine you want it to last a little bit longer. Some you may want it to be biodegrading faster. But as I said, I think the future is really this therapeutic carrier design that I said over and over again, that we would be able to deliver therapeutic cargo at the same time. So then it would dictate the lifetime of this nanoparticle, which would be influenced by how long a sustained period one would like to see. But the short answer, I think it should vary from application to application.
So there's a question from Zhongming Li. Rheumatoid arthritis is a nice model for systemic inflammation. How about application of nanoparticles for other localized inflammation for soft tissue, such as example tendons and muscles?
Zhongming, thank you. So that's a good question. That's why the second example I show is that I use cationic microparticles. So then it will be applied locally. We had in mind to use that for osteoarthritis. So then it could act in a local manner. For your application for tendon and muscle, again, microparticles applied locally would be better. Now, then I don't have to worry too much about the systemic toxicity of the cationic structures. in terms of tendons, so for example, one could even think about in the form of a cationic fibers that could be applied there.
Okay, there's a question from Si Guang Yang. Thank you for your presentation, Professor Leong. As you introduce microparticle, can be used in localized scavenging. So if you apply that in impact wound healing in animal model, how to determine the lifeline given that early inflammation is important?
Okay, that's right. That is a tough question. I could speculate, if you really want to try, you might want to try a laminated structure, that the outer surface area, that the outer surface layer is not so cationic. And for example, one could imagine the outer surface layer would break down in a matter of days, then the scavenging could happen. That's what I would advise if you really would like to try that.
Okay. Yeah, so Barbara has another question. Cam, how would you scavenge and deliver at the same time through different functionalization of the nanoparticle or delivering stuff encapsulated inside the nanoparticles while scavenging by absorption on nanoparticle surface?
Thank you. That's a good question. It has been done. It's very doable. We have that right now in the lab. We applied that to a cancer, to a breast cancer model. Now, if you remember the cationic nanoparticle structure, it was a diblock copolymer. PLGA is one block, and cationic polymer as the other block. So the PLGA, that lipophilic hydrophobic segment, would be able to encapsulate lipophilic drugs. And that's what we have done. Then we would have kind of the same cationic nanoparticle, but with drugs entrapped in the core. And there would be sustained release of the drug from that kind of a cationic nanoparticles. Your hunch is correct. Then it would be difficult for us to encapsulate a hydrophilic drug or a polar drug. Then it would be difficult. But for lipophilic compound, it's not a problem.
Yeah, so Alan Lee has a question. Thank you, Dr. Leong, for the inspiring talk. Could you comment on the use of nanoparticles for the modulation of macrophage polarization and phenotype?
Yes, we have done a lot of that. Cationic structure already, we have shown that we could switch the M1, M2 phenotype right now. But that may not be the most efficient way of doing. Many other groups have done that by encapsulating particular compounds to do that. And so you might want to check the literature on the kind of compounds that people use to do that.
Okay. I think there's one last question. Su Wan Ding, out of curiosity about the behaviors of polycationic materials in blood vessels, assuming they are injected, besides the possible aggregation, are there observations indicating that negatively charged proteins such as serum albumin may adhere to these particulates?
They should. Now, that's also kind of the hand-waving opsonization process. Any foreign materials come into contact with the biological structures would have interaction with the proteins in the body that way. I do not have a firm answer on that, but I'm sure there is some. How dynamic interaction, I cannot say. Now, what would it do to the blood vessel? So if you remember early on, I showed, maybe in the middle of it, there's a phenomenon that people are seeing, that nanoparticles would lead to leakiness in the vasculature. That's called the nanomaterials induced endothelial leakiness. That is affecting either one of the junction proteins there. So nanoparticles would make the vasculature more leaky.
Okay. I think that is all the question. That was excellent. I think there were many questions, a lot of interest in your talk, Cam. Okay, so I think that's all the time we have for today. We make also very good time. I would like to thank again Professor Leong once again for your very nice and fantastic talk, and also thank you all of you for attending today's WACBE webinar. Be sure to register for our next webinar on February the 27th. And enjoy the rest of your weekend, and also Happy New Year. Okay, bye everyone. Thank you.
Thanks. Yeah, thanks, Cam. Thanks, Savio and the CT. Yeah, Happy New Year.
Thank you. Bye-bye.
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 Bye.